talk a little bit about bilirubin. Let's try to build our concept about bilirubin because it's one of the most basic thing that you can study in medicine. So the concept of bilirubin, rides along jaundice, right? If you want to really understand jaundice well, we must understand what the hell bilirubin really is, right? So the concept of bilirubin starts with lysis of this RBCs, right? RBCs are these, RBCs are red blood cells of our blood, right? They carry oxygen. That's their primary function. They are anucleated. They don't have any nucleus. It means they cannot produce their daughter cells, so no daughter cells of RBC because they are anucleated. Other thing is that their size is around 3 micron, and that leads to them being able to travel in small arteries. So that's very important as well. So the starting point is that old RBCs, they die off, and 1% of RBCs every day they die. And when old RBCs they die, it leads to their death, of course. And what really RBCs, let's try to understand what RBCs really is, right? Because it's a general term, RBC, red blood cells. Why is it red? Why is it present in blood, and what's its function, right? So RBCs is made up of hemoglobin, and this hemoglobin has two parts. It's had heme and it has a globin part in it, right? So the heme part gives the color, right, the red part, and it's the pigment, whereas the globin part is a chain that allows binding of oxygen and allows these functions. Okay, so globin is made up of amino acids, and heme is made up of two things, which is succinyl CoA and glycerin, whatever you want to say. So both of them are amino acids. So basically it's a protein. Okay, it's amino acids, so it's protein part. So heme has a ferrous part and a porphyrin part. Now ferrous is iron, and porphyrin is like a metal, it's like a metal claw that holds this ferrous with it. Okay, if we have problems in porphyrin, we will have we'll have iron deficiency. Okay, so we must have good synthesis of porphyrin so that we can hold iron, and if we are able to hold these two things, we will form this heme. And if we are able to join globin chain with heme, we will have hemoglobin. So that's the concept about hemoglobin, and that's how it is able to hold oxygen with it. So let's get back to bilirubin again. So when our RBCs they die, they die in spleen, liver, and bone, right? And why they even die at spleen? Why? That's a good question to really think. Why do they die in these weird places? Spleen, liver, bone. They can die just at your hands or maybe in your brain. Why they die at spleen? Because spleen, the number one function of spleen is to filter blood continuously. Old damaged RBCs that are not so flexible because they have lost their their function of being very. So the thing is that RBCs have this biconcave shape. They have to be in this shape. If they lose their shape, they are tagged as dysfunction, because they cannot be, they're not really flexible now. So that's number one problem. Once we feel that these RBCs are not able to hold their position, hold their shape well, we need to throw these RBCs. So spleen is number one organ. So we have something called as macrophages in spleen. Now what is macrophage? Macrophage is just a big cell that can phage. What is phage in medicine? In medicine, phage means digestion, right? So this macrophage actually works by, you guys must have this thing called bleach at your home. So macrophages uses bleach to actually destroy thing in your blood. So macrophage, they come up at this spleen, and we have these splenic macrophages. They engulf and they destroy these RBCs, right? And that's why the spleen is the primary site of RBC death. What about liver? Well, liver have something called as Kupffer cells. These Kupffer cells are also macrophages, just we name them differently. They also remove these old RBCs, right? We want some really healthy, flexible RBCs so that we don't have any sort of clot or any sort of thrombus formation, right? And what about bone? Well, actually it's not bone, it's bone marrow. It's a minor site. Most of the lysis of RBCs, it happens at level of spleen and liver, which we call it extra medullary. And if the lysis of RBC is happening in the bone marrow, we call it intramedullary. Okay, so that's another concept. Really try to understand it. So after, what happens is that this end product of RBC, when RBC is destroyed, we get heme back, we get globin back, right? And then heme gives us iron, and then we have something called as bilirubin, right? And this bilirubin, right, this heme is coming from this bile. This bilirubin is coming from bile, right? So this iron that is produced by death of RBC is reabsorbed back, right, with the help of liver. Liver is like a master regulator of enzymes. It can produce a lot of enzymes that can reabsorb iron back inside our body, because iron is a luxury. Always remember that. Whereas porphyrin ring, right, now these porphyrin rings are super toxic, right? And it's a good concept to understand why they are toxic, why they are super toxic. Now really understanding porphyrin rings are super toxic, it works via multiple ways. First of all, they are really good at making or generating these OH, that are free radicals, right? And now these free radicals can actually produce a lot of enzymes, and they can phagocyte your protein. So that's really bad for you. So they have something called as reactive oxygen generation, right? Other thing that porphyrin ring can do is that they can actually damage your membrane, right? So these porphyrin are something called as lipophilic. It means they love the fat. They can slowly, easily can pass through any sort of lipid membrane. Once they insert themselves into the lipid membrane, they cause oxidative damage again, right? The oxidation part. Other thing that they can do, porphyrin, is that they have something called as photosensitivity, right? What happened here is that when porphyrin that accumulates on your skin, right, and you are exposed to sunlight, what happened here is that this painful blisters, right, they are formed, right, and it causes skin damage. So this is one of the classical features of this porphyrin accumulation on skin, right? So it must be removed by your body. It's toxic as heck, and we need, because it can do oxidation. So body needs to break these hemes safely. Heme give us porphyrin, right? As we know, heme is made up of ferrous and porphyrin. Ferrous is the iron part, porphyrin is the metal claw that holds this iron, right? So we need a safe mechanism to to just throw this porphyrin away, right? Because it's dangerous for us. So what happened here is that this is where the bilirubin is found, it's used for. It detoxifies. How? Because it neutralizes these free radicals that are generated by these porphyrin rings, and this happens between the tissues, right? As bilirubin is the unconjugated bilirubin is fat soluble, so it can travel between one tissue to another tissue, right? I will talk how it's fat soluble and how it's water soluble. We'll talk about it. What happened here is that this bilirubin actually detoxifies, right, by neutralizing these free radicals. So at low level, bilirubin is actually very protective, right? But at high level, bilirubin can actually one of the biggest problem in neonates. So we try to make sure that we have less levels of bilirubin in neonates. Let's try to understand about this bilirubin more. Let's try to make this concept a little bit tighter. So the pathway how bilirubin is made. Okay, so we have heme, and when heme breaks down, we get something called as biliverdin, right? Now this biliverdin, right, this is made inside a macrophage, and these macrophages are present in the spleen. So I want you to understand the location where it's being made. So when heme breaks, we get biliverdin. Now this biliverdin gets further breakdown into bilirubin. Again, this is happening in the macrophages of these spleen. We call them specialized splenic macrophages or anything like that. So after that, what happens is that this bilirubin, right, it is still unconjugated. What is unconjugated? Well, conjugation means soluble. What soluble? Water soluble. So if I'm saying unconjugated, it means that it's insoluble in water. And what I mean here is that it's really soluble in fat, right? We have two types of things in our body. Either they are hydrophilic or they are hydrophobic. they love the water or they hate the water. If something is hydrophobic, it will not be excreted in the urine. You need to have hydrophilic elements, hydrophilic compounds, hydrophilic metabolites. What are metabolites? They are just end product of a metabolic reaction. We call this metabolites, right? So we need to make sure that molecules and the end product and the waste product that your body generates, we need to make sure that they are conjugated, so that they are water soluble, so that they can be removed by your kidneys. And this conjugation is usually done in liver by hepatocytes, right? What are hepatocytes? Hepatocytes are just the cells that are present inside the liver. So that's the concept right here. So this bilirubin, it binds to albumin, right? And why do we even need to bind this bilirubin, this fat loving unsoluble, water insoluble form, to bilirubin, to albumin? Why? Because albumin is a safety latch, because this bilirubin is actually very toxic. It is fat soluble. It means it can freely travel between tissues, right? And if it's really free with these, because why it can freely travel between tissues? Because our cells are made up of bilipid layer. It means there are two fat layers on the periphery of the cell. So our cells are protected by these fat layers. So if something is made up of fat, of course it can cross. So that's the concept right here. So bilirubin, it can cross. So once it binds to this albumin is a protein in nature, right? So this complex of fat and protein is made. Now once this complex is made, this cannot actually cross this bilipid layer of our cell. So that's how we keep the bilirubin safe from tissues, right? Because most of the tissues can, they have this bilipid layer, and once bilirubin is unconjugated, it can cross freely and it can be very toxic, right? It can create amazing levels of these free radicals in our tissue, and free radicals can further they can just destroy a lot of things. So I don't want to go into how free radicals destroy things, because that then we will just deviate from the main topic, that is bilirubin. So bilirubin binds with albumin, and then it can travel to liver now, right? Because albumin are these protein in our blood, right? And they are just carriers of albumin, right? And albumin is one of the carrier for many things, right? Okay, after what happens, that conjugation thing happened, right? Conjugation process itself happens, right? And this conjugation happens in the liver, right, at hepatocyte, using an enzyme called UDP, glucuronyl transferase. Okay, my naming is very bad, but please pardon me. So it adds something called as glucuronic acid, and if we have any sort of deficiency in this enzyme, we will have this thing called Crigler-Najjar syndrome. And what happened here is that we have less levels of these enzymes, so conjugation is not going to happen. We're going to have insane amount of bilirubin, unconjugated bilirubin, and this bilirubin is going to go to your brain, and then we'll have something called as connectors. Now this connectors is one of the syndrome. It's one of the situation where you have insane amount of bilirubin in your fat tissues of your brain. Now most neurons are covered by a sheath, a layer of fat, so that it is insulated. The current inside the neuron is insulated. We want a fat layer of fat layer on the neuron, so that any sort of short circuit, any sort of abnormal communication between two neurons is not happening. That's why we need fat on the neurons, right? So what happens here is that this bilirubin goes and it binds with these fat myelin sheath on the neurons, and then this oxidation that is created by bilirubin, mediated by bilirubin, actually destroys this myelin sheath. And once the myelin sheath is gone, you will have abnormal levels of conduction between two neighboring neurons. So that is how the neurotoxicity happens. Okay, so conjugation happens at liver, and we know about UDP, glucuronyl. Okay, so it's a long name. It's a transferase. I usually call this enzyme GG. Wait, let me. Okay, where is this enzyme name? Wait. Okay, just bear with me. UDP, glucuronyl transferase. Okay, it adds glucuronic acid. That's very important. I'll tell you how. Okay, so we are at this point of the story where the conjugation has happened, right? The liver has made this unsoluble, very bad unsoluble bilirubin into this soluble, urine loving, water loving, bilirubin. Now this bilirubin is safe to, pee, it's safe to urinate, right? Safe to excrete, right? So that's like that's the story we are here right now. Now I want to go a little bit deep inside this conjugation, what really is conjugation, how it really happens. So unconjugated bilirubin, right? So this unconjugated bilirubin, why it's even fat lover, why it's even fat soluble? First question is this. Second question is that why it's a water hater molecule, right? Why it's something called as hydrophobic. Why it's even hydrophobic? That's a basic question. We should understand that. To understand that, we should know about the structure of bilirubin, right? So the molecular structure is present in the way that the polar groups, the ionized, the charge group is inside. It's hidden inside the structure. It's like a banana, right? So this is a banana, right? I'm making a banana, and this is how it really is, right? So the polar structure is right here. It's inside. And usually what happens is polar group, it's usually responsible for the binding with H2O, right? The hydrogen bonding happens with the polar groups usually, but this is what happens. So this polar group is hidden inside, and the hydrophobic end of this molecule, of this structure, is outside. So that's why unconjugated bilirubin is hydrophobic. So I have answered your one question. Another question is why it is lipophilic, why it is a fat lover. So if any molecule has either something called as non-polar bonds that are non-ionic, right? Non-polar bonds. I hope you understand non-polar bonds. If you don't, please Google it. It's really important to understand. Other thing is if it lacks exposed ionic bonds, right? So if both the conditions are met, then we will have a fat lipophilic or a fat lover molecule. So that's why unconjugated bilirubin is lipophilic and it's hydrophobic. So that concept is done. Other thing is this, what is really conjugated bilirubin, right? So what here happens is that in hepatocytes we have something called as smooth endoplasmic reticulums, right? These smooth endoplasmic reticulums are the storage sites of a lot of enzymes, right? So inside this smooth, why is it even smooth? Why is the naming is even smooth? Because they don't have these granules that are present on the endoplasmic reticulum, right? Now what is endoplasmic? Endoplasmic means inside the endo means inside, plasma means the plasma of the cell, the cytoplasm of the cell. So they are present inside the inside the cell. And smooth means they don't have any granules of protein producers, right? So they are these smooth endoplasmic reticulum body. It's like a body, it's like a cavity, right? And what they have is that they have this UGT1A1, this enzyme, this same enzyme, UDP glycoronyl transferase enzyme. Now what happened here is that it adds this glycuronic acid, right? Now we know that once acid is added to almost anything, it becomes water lover, right? It becomes hydrophilic. Usually this is the case with even protons, right? What are protons? Protons are these H positive ions that are free to just cause havoc, or maybe free to cause destructions, right? So once we add this glucuronic acid, this bilirubin becomes something which is called as mono, or it can become a diglucuronide, right? Now this glucuronide in nature, it's a sugar, and now after adding this acid, right, it has an OH end to it, right? So it's a sugar with OH end. So what happened here is that with the help of this OH, the closed structure of this unconjugated bilirubin, it opens up. Once the structure opens up, the closed structure opens, and then unconjugated bilirubin opens, and this polar end that was otherwise hidden inside the structure now is exposed. Once the polar group is exposed, now this polar group that is positively or negatively charged, once this is charged, this can bind with water, and it binds with the help of hydrogen bonds, because water is H2O. So polar groups now can join with water. So this is important, the structure. So that's why it's indirect. This is indirect, this is direct. I hope you can remember that. Okay, so we understand this we understand this bilirubin, right? Little bit we understand, we have an idea about this. Other thing is that let's try to understand how the bilirubin really affects our body. So what happened here is that this albumin, right, the protein that carries these unconjugated bilirubin, right, the albumin is this safety lash, right? It carries this. Make sure that this free unconjugated bilirubin is not free to cause damage in these fat cells, right, that it can, cross this bilipid layer, right? So once albumin is attached with this bilirubin, it can't cross this bilayer, right, because it has a protein end on it, right? So protein cannot, they cannot jump these bilipid layers, right? So here, what happened here is that only unconjugated bilirubin deposits at tissues at it as it's fat soluble. So that's I'm just saying same thing again and again. So if anything that can cause, that can lower the amount of albumin in your blood levels, right, it can cause damage via unconjugated bilirubin, right? Because bilirubin in its nature is really toxic, right, in high quantity. If it's present in low quantity, it's like antioxidative, right? It is against oxidation. If it's present in less, quantity, if it's present in high quantity, it's very oxidative. So anything that can cause less albumin, it's gonna lead to damage via this abnormal levels of bilirubin. Okay, after what happens is that UCB, the unconjugated bilirubin, it is a fat lover, it's a lipophilic substance. So why newborns are at risk? Here what happens is that a preterm baby has less function of UGT1 enzyme, this enzyme that converts UDP glycuronyl transferase. This enzyme is low in prenatal child, prenatal neonate, because they are still developing, and once they have less amount of this enzyme, they will be able to produce less conjugation. If there is less conjugation, there will be slow clearance of this unconjugated bilirubin, right? Because this, I hope we understand this, why the low clearance is happening, because there is less conjugation happening. Once we have less clearance, we'll have high levels of bilirubin in our blood, in our child, in our preterm babies, right? So that can lead to problems. Other thing that can happen is that this blood-brain barrier called BBB, it's a barrier between your blood system, artery system, and your brain. Your brain is not exposed to every chemical that you put inside your body. It is very selective. Neurons are like ladies. They want to stay alone. They don't want your interaction. They want to make sure that they have completely isolated environment. They don't want to, talk to every stranger, right? So blood-brain barriers, what happened here is that, right, because they are a preterm, because it's a neonate, they have weak podocytes and weak astrocytes. Now both of these are the constituents, both of these, they make blood-brain barrier, right? They are the forming tools. It's like a raw material for your blood-brain barrier. If you have weak podocytes and weak astrocytes, of course this bilirubin will be able to cross this blood-brain barrier that your baby has. Now what happened here is that due to less function of this enzyme called UGT1A1, the level of bilirubins will be high in the plasma. If your body senses the levels of bilirubin are insanely high, a systemic inflammation will start to happen, right? A lot of systemic inflammation leads to this thing called so we have venules, right, and we have arteries. So arteries have something called a smooth muscle. So this is the reason they can dilate and constrict, because they have these smooth muscles. Whereas this venules, or maybe these capillaries, or vein blood, right, veins, they have these fenestration, these windows. So what happened here is that in systemic inflammation, the windows, the size of windows, they open up. So we have more permeability, right? So what more permeability means, the blood-brain barrier gets a little loosed during systemic inflammation, right? Once the inflammation starts, right, we have weakened BBB. Now blood-brain barrier, weaker blood-brain barrier. Now this inflammation takes place via microglial cells. These are the neuronal immune cells. Okay, just know that these are just cousins of macrophages, or even you can call them neuronal macrophages, you can call them that. So this happens, weaker blood-brain barrier, so the bilirubin can now cross the blood-brain barrier. Other thing that can happen, that in child, the neurons have a very thick myelin layer, right? And once this thick myelin layer is exposed to bilirubin, high amount of bilirubin is binded to these myelin, right? So more deposition of bilirubin happens. All of this leads to this general function. What are these general function? We know that mitochondria is needed to produce ATP, right? And so the function of ATP stops, the function of mitochondria stops. Now we know that huge amount of bilirubin can lead to pro-oxidant, right? And what really happens in details here is that this bilirubin it stops the antioxidant levels of, we have these neutralizers that are already present in our body that can neutralize these OH protons, or H plus protons, or OH minus free radicals. They just bind with them and then the neutralization happens, right? So what happened here is that this bilirubin actually decreases the levels of these antioxidant molecules. Other thing that can happen is that they can stop the DNA RNA synthesis, so we can have the shape of RBC will change, the size of RBCs will change. So we'll have this problem in the synthesis of RBCs and even synthesis of all cells. So it can be pro-apoptotic, because if we have bad shaped cells, again a system called P53, this is a system, right? And now this system is present in your body. It makes sure that any sort of badly made cell, any sort of any cell that is formed in a bad manner, that is supposed to that is not supposed, for example, a car is supposed to have 4 tires, right? Imagine your car has just 2. Now it's a bike, it's not a car. So what will happen is that this system, P53 system, right, that is present in your body, is gonna recognize that and is gonna say, okay, remove this bike. So it can be apoptotic, so you're gonna have a lot of death of cells. Now remember that apoptosis is ATP dependent and necrosis is not really ATP dependent. So that's the difference between necrosis and apoptosis. Apoptosis always requires energy because it's a systemic approach to remove defective cells, where necrosis is usually like a murder of these cells, right? So this can happen. Other thing which can happen is that they can stop the production and uptake. Production is that production, right? You produce, and uptake is that, imagine, okay, I'm gonna explain you this neurotoxicity very simply. So we have 2 synapses. We have one synapse. I'm sorry. So this is right. This is one. So this is a synapse. This window is a synapse. This is a pre-synaptic neuron. This is a post-synaptic neuron. Usually what happens here is that we have any sort of neurotransmitter. For example, I'm going to take acetylcholine. Acetylcholine, called ACH, is a neurotransmitter, and it's a excitatory neurotransmitter. Usually what happens is that when we produce acetylcholine, it leads to increased level of CAMPs, and then we have, for the pathway, what happened here is that in short, acetylcholine can excite your neurons, right? It can depolarize your neuron. What do I mean by depolarization? Usually what happened here is that neurons usually sit at minus millivolt. That's their resting membrane potential. We need to put some positively charged ions in the neuron, so that the resting membrane potential, the potential across the neuron, it rises up. And once it rises to a certain potential, these voltage sensitive with these voltage sensitive channels or gates, whatever you want to call them, they open up, and then we have a influx. Then we have these other molecules come inside the play, right, which is acetylcholine. So acetylcholines, right, so let's get back to this neuron, post-synaptic and pre-synaptic, right? So this is a synapse. What is a synapse? Synapse is just a communication between 2 neurons. That's synapse. So this is a pre-synaptic and this is a post-synaptic neuron, right? So this is a post-synaptic surface, this is a pre-synaptic surface. Acetylcholines get released here, and it goes and it attaches here. Some sort of function what acetylcholine is supposed to do, right? I'm not gonna go inside because acetylcholine can lead to many functions. So acetylcholine binds at this post-synaptic surface, and then we have these functions that's going to happen. After, what happens that acetylcholine goes back inside the presynaptic neuron. What can happen here is that this bilirubin, right, it goes and it binds with this uptake system of acetylcholine. So here what happened here, so acetylcholine will not be able to go back, right? Once acetylcholine is not able to go back, it will go back again, and it will trigger this whole reaction, whole pathway again. So we will have something called as hyper reactivity, right? Now, a cool thing, amazing thing here is that a child with chronic means long time, chronic hyper, hyper means a lot, bilirubin will have hyper reactivity. The child will be really irritated, because he is experiencing these abnormal levels of acetylcholine. But a child that has acute means it's a new phase, right? It's a new disease, right? So acute hyperbilirubin, the child will be hypo excited. He will be really depressed, he will be really he'll be really lethargic, in other words. Now I want to really explain that, because hyperbilirubinemia in the brain, right? So in acute phase, right, we have something called as encephalopathy, right? Now here, what happened here is that it's just an edema of your brain, right? And this bilirubin, it starts to affect the brain, right? The baby become really lethargic. It's like a floppy baby, right? It's hypotonic. What is hypotonic? Hypotonic means less. Tone means tightness in the muscle. So it's hypotonic, poor feeding, weak cry. Sometimes he whatever. It is crying, right? It's even vomiting. So the reason is that the early toxicity depresses the neuron activities, right? Because the acetylcholine, which is not getting uptaked, right, it leads this negative feedback on neurons, and they get super depressed. It's like a safety mechanism for you. Okay, so I hope we understand. That's why, because of the negative feedback loop, because neurons have this safety system, that if we have hyper levels of these neurotransmitters, the neuron will be really depressed. It's like a safety mechanism. But in long term, it leads to this accumulation of acetylcholine. Okay, how really it happens? Okay, imagine we have this bilirubin that has already binded with this presynaptic neuron, right? Here it has already binded, and this is my post synaptic neuron, right? And this acetylcholine it cannot go back, right? So here we have less acetylcholine. So right now we have less activity of neurons. It's hypo excited, right? It's not really excited, it's really depressed, because we have less amount of acetylcholine. But with time, there will be accumulation of these acetylcholine. So instead of one acetylcholine, we're gonna have like 15 acetylcholine atoms here, and then this 15 is gonna right away act on this neuron, and then we'll have hyper excitability. That's why in acute phase the baby is floppy, and in chronic phase the baby is really irritated, it's really agitated. So that's really amazing to understand. I hope we really make our concept really clear on this thing. So there is no safe dosage of bilirubin in our babies. Our babies have different system, everything is different, so it's really hard to say how much is safe for someone. So we have these clinical symptoms based on these activities that's happening. Other things that we can actually do things, treatment for these things, is that phototherapy and exchange transfusion. So exchange transfusion is that we just exchange the plasma of the defective baby, the defective prenatal, with a healthy baby. It replaces the albumin, which is the prime carrier of bilirubin and RBCs, so that we don't have hemolysis, right? And other thing that can happen is that phototherapy. We're going to expose the prenatal baby to these blue light. What happened here is that these blue light, they convert the unconjugated, the fat lover bilirubin, into this conjugated, the hydrophilic, the water lover bilirubin. And it's not really converting, it converts this unconjugated into conjugated isomer. Same, I hope we understand the difference between isomers, right? So I don't want to explain isomer because okay, after this story, this story is not ended yet. We must understand what is something called as porphyrin, because it was mentioned in a story, right? And we'll understand why it's yellow, why it's not, purple, green, right? So here, what happened here is that let's try to understand something called as porphyrins, right? I already told you porphyrins are these metal claw that hold this ferrous in porphyrin ring. What is this porphyrin ring? Now, porphyrins are called shell-eating molecules because they are these metal claws. They can bind the iron in it, right? Now, 4 pyrrole ring joined together with methynyl links, it gives us something called as porphyrin, right? So what happened usually here is that 4 nitrogen atoms, they sit inside the center of this ring, and they are the reason we are able to hold this metal ion in it, right? That's how we can hold ferrous in it. Okay, so erythroblast, what is erythroblast? Erythro means red, blast means something that can produce. So these are precursor cells of RBCs. They make RBCs, right? So these cells, they make RBCs and they make these pyrrole rings, right? So we have these pyrrole rings, 1, 2, 3, 4 pyrrole rings joined with methynyl links together, right? And this is called protopyrrole ring. Once we put iron in it, right in the middle, we get heme. Now, if we add globin chains to it, right here, we get something called as hemoglobin monomer. Okay, and we have 4 types of hemoglobin, by the way. HB are 4 types, so just you should know that. And then we use this protoporphyrin, something called as protoporphyrin 9. It's a subtype of protoporphyrin, right? There are multiple types of protoporphyrin because protoporphyrin are found in animals other than us. They are found in in fishes. They're found in plants, right? So we should know that we use protoporphyrin 9 to hold iron in it with the help of nitrogen. Okay, so other thing that can happen to this protoporphyrin ring is that we can add magnesium in this ring, and then we'll get chlorophyll, and that's how we get this chlorophyll inside the plant. So another cool thing, right? And usually this chlorophyll is the reason why the plant is green, because protoporphyrin actually is a light absorbing substances. It absorbs certain light, it absorbs certain frequency of light, and usually what is not absorbed is reflected inside your eyes, and it goes inside your retina through your iris, and it goes inside, and this retina actually has receptors. It has these receptors on the periphery and these receptors in the middle, in the center. So these are peripheral receptors and these are central receptors. I don't want to go inside your ophthalmology physiology, but this is how you are able to see colors. The colors that are absorbed by the substance are not seen, but the colors that are reflected back to you are seen, right? That's how it works. So protoporphyrin ring, if magnesium is added, produces green color, that is chlorophyll. If cobalt is added, then we get something called as B12. That's amazing, right? Now we have B12. So I hope we understand this concept. Other cool thing that protoporphyrin can get is that this is the reason why we have, by the way, protoporphyrin, this is the reason why we have red color, right, in our blood. And same as heme is also made up of glycine and succinyl CoA. And this is the first step of heme production, and this is the same production line for porphyrins as well. So glycine plus succinyl CoA can give us porphyrins. So if we have problems in the synthesis of heme, usually what will happen is that we'll have problems with the production of porphyrins. So these things go hand in hand. And by the way, why is bilirubin yellow? I'll tell you about it. Wait, before that, what happens after conjugation is that this conjugated bilirubin usually is peed out, right, inside a urine, right, excretion of conjugated bilirubin, right, with the help of bile. With the help of bile is produced by liver as well, and it's stored in gallbladder. And we have these enzymes called a lot of enzymes that can contract gallbladder. It can contract gallbladder, and then we have production of bile. It comes inside our intestine, and with the help of bacteria in our intestine, bile and bilirubin, they are both joined together, and then we have we have a formation of urobilinogen, right? Now this urobilinogen leads to formation of stereochobalin that is produced that forms a lot of feces. That's the shit you take. and it actually, this urobilinogen actually makes urobilin. That is the yellow part of your urine. That's why your urine is yellow, and that's why your feces is, that's why your shit is brown, because of the formation of urobilinogen, because it has a bilirubin part in it, right? A conjugated bilirubin part. What other question here, what other question that can happen here is that why is it even yellow? Because biliverdin, the form before bilirubin, right? So heme makes biliverdin, right? So the biliverdin actually has long conjugation bonds, right? And different conjugation bond, the length of conjugation actually can change the color, right? We see. So usually what happens is here is that this long conjugation bond can absorb red and oranges. Once it absorbs red and oranges, the green color is usually reflected to us, and that's why the biliverdin is greenish in color. After reduction, right? After reduction, the biliverdin turns into bilirubin, right? In the macrophages, right? So what here happens is that the formation of bilirubin, right? These have shorter, conjugation bonds. Now these shorter conjugation bond, they absorb blue and violet light. Once they absorb that, the yellowish light is reflected to us, and then we see that bilirubin is giving this yellow tint on the skin, on the sclera, and wherever there is some sort of fat present, you will have bilirubin there, because bilirubin is unconjugated, especially unconjugated bilirubin is fat soluble, right? So that's why bruises, right, changes color. Bruises are these wounds, right? I hope you understand that. So the acute color of the wound is red and purple due to biliverdin, right? Because red purple leads to greenish biliverdin, and biliverdin is green. So the color change from red purple goes to greenish in our wound, in our bruises, and then the color of bruises changes to yellow because of the presence of, because biliverdin is getting converted into bilirubin, because macrocytes are also present on the site of bruises, right? Because bruises can cause inflammation, and inflammation calls macrophages. So that's why the color of this bruises changes. So that was the whole concept about bilirubin. Why we need that? We need that to detoxify, to fight against, the oxidation of porphyrins, because these porphyrins are carrying this Fe. So these rings are really dangerous for you. So we need to stabilize these rings using bilirubin, and if we have a lot of bilirubin, it can actually be dangerous for our own self. So that was the whole concept of bilirubin. I hope you have a nice day.